Welcome to your first real lab in biology. Yes. Before you ask me the most popular question when beginning a lab, which is, Miss Rune, do we get to pick our partners? I'm just going to answer that right now. Here's the answer. No. Here's why. Pleased to meet you. I am your lab partner. So I figured it might be nice for our first real lab. We are going to work together, you and me. And the way that's going to work is that I'm going to record myself doing this lab in Pivot. That's what this is. And you're going to work along with me. And that's going to help you get a sense for, like, how does this software even work? How do I take data from a video? How do I graph the data that I take? So let us look at this together. We're up here in Pivot. Hopefully you're here too. And we're working on this Exploring Photosynthesis Part 1 lab. So this lab tells us we're going to investigate the changes in gas levels. We're talking about oxygen gas and carbon dioxide gas for plants when they are exposed to light and when they are in the darkness. So Part 1 has this nice video. It says that we've got these plants in a closed container. That's this thing. And there's gas sensors that are going to measure carbon dioxide gas in parts per million. So that's literally like, I've got this carbon dioxide molecule. If I've got 1 million molecules of air, how many of those are carbon dioxide? So parts per million. And then the amount of oxygen gas. Now there's a whole lot more oxygen gas in the air, so that's going to be a percent. So that's like parts per 100. That's what a percent is. So our goal is to understand how these gas levels change in different conditions, the conditions being light and darkness. So come with me, let's enjoy this video together. You're going to notice that there are some things you can change. So when you're in this interactive, you can click this little tool symbol, and that pulls up a stopwatch. So you can click your stopwatch, and look, it's a stopwatch. It's available to you. I'm going to drag it here so that I can actually see my stopwatch and my gas meters. There's another thing I can click down here. So if I click on the leaf color, it pulls up this option bar. And my leaves can either be green, like they are already, or they can be red. Now if I click red, then when I load, I got to click load, it's going to be a different video that plays. And this time, get ready. Oh my gosh, it's a red plant. Now if I had green, load, make a prediction. Ah, it's a green plant. I think that's a basil plant, actually. So notice you can change some things. And the first piece down here is our instructions tell us, select your stopwatch, did that. And then we're going to advance the video to the beginning of the time-lapse portion. That'll make sense. Don't freak out. And then we're going to reset our stopwatch so that it reads 0. And we're going to start the video and record our data about the light, carbon dioxide, and oxygen. Okay, so this instruction writer person doesn't want to tell us a whole lot about how to record the data. So I'm going to help you. I'm going to sort of give you some clues about how a scientist, me, and now you, would record this data. So first of all, it tells us we want to advance the video to the beginning of the time-lapse portion. So let's play until it says, we're closing the container, time-lapse. Okay, so I paused super quick. Now it said, reset my stopwatch to 0. Reset. Okay. And so now I need to think about a few things. How am I going to set up my data table? How often am I going to collect data? This says every 10 minutes. So I need to be watching that stopwatch and pausing the video at 10 minutes, at 20 minutes, etc. Now the things I'm going to have to think about are, is it light or dark? It'll be obvious. What is the CO2 level in, my units are ppm, and what is the O2 level in, my units are percent? So if we're at 0 minutes, we can already collect this data here. So let's scroll up at 0 minutes on my stopwatch, not down here on the video, but on my stopwatch, I've got CO2 level, gas 1, is 731 ppm. My units are built in here. So now I need my O2 level. My O2 is 19.62%. I'm typing it. Boom. Okay. Data point 1 collected. All right. So now I am going to press play, but only until my stopwatch says 10 minutes, right? Because that's my next data point. So play. Oh, I'm close. Am I close enough at 9.5 minutes? What do you think? I think so. The chances of me pressing play and pause and getting it to exactly 10 minutes are pretty weak. I'm not that good at clicking. So I'm going to take my data point now. I'm close enough. So my CO2 level right here is 641 ppm. 641. I realize I skipped a column. I need to go back up and make sure, yep, it's still light. So I'm going to write that down. And then my O2 level is 19.66%. 19.66. So now I'm going to play and I'm going to pause again at 20 minutes. This goes fast, guys, so warm up your clicking fingers. Whoop, I kind of missed it. Does it matter? Not really. But let me see if I can pull this bar back to get it just a little closer to 20. Nice. Okay, it looks like I can either get 19.9 or 20.1, but that's good enough for me. So I've got a ppm carbon dioxide level of 564. 564. It's still light. And my O2 level is 19.68. Pause, think with me here. Does it make sense that, like, both these levels are changing, right? My CO2 looks like it's going down. My O2 looks like it's going up. Why is my CO2 changing, like, by the 100? Like, I go from 700 something to 600 something to 500 something. But my O2 level is like 19.62. 19.66, 19.68. Why does it look like that's changing so little when the CO2 is changing so much? Think about that. Pause, think, press play when you want to hear the answer. So remember, a percent is a way bigger amount than a part per million. So it makes sense that the parts per million are flying and the percents are just, looks like they're barely changing, but we got to keep in mind that those are different units, right? Okay, sweet. We thought about that. Go us. All right, so let's play and then pause at 30. Pause at 30 minutes. Remember, I'm looking up here for my time. Play. Pause. Ooh, nice. Go me. I'm clicking like a champ. So 30.1 minutes, I've got 501 ppm. Is it still light? Yeah, it's still light. And what's my O2 level? 19.69. All right, I'm doing it again. Play. Oh, it's dark. I overshot. I'm going to go back. There we go. Okay, so now at 40.1, it is dark, so I need to make sure I record that. It is dark now. I've got 550 ppm for my CO2 level. And my O2 level is 19.6. 60 to be precise. All right, let's get our 50-minute data. You guys should be getting a hang of this now. If you're bored of me, just turn me off and take your own data. So we've got 613 ppm. Whoa, something changed there. We'll talk about it. Still dark. Hopefully you noticed we're going up now. And what's our O2 level? 19.54. Things are changing. Let's get, whoops, didn't mean to do that. 10 minutes more till 60 minutes. Oh, lights turned on. It's 666 ppm, so light 666 and 19.52% oxygen. All right, 10 more minutes. We're going to 70. Oh my gosh, guys, look how good my clicking is. Okay, 605 ppm. All right, things are changing again. Hopefully you're noticing a trend. It's light now, and it looks like our carbon dioxide is going back down. What's my oxygen level? 19.58. All right, 10 more minutes. If you're getting bored of this, pat yourself on the back. That means you're getting good at it. Okay, so 535 ppm is my CO2 level, and my O2 level at 80 minutes is 19.62. And it's still light. Ten more minutes. All right, close enough. At 90, it's still light. 483. Is my carbon dioxide level, and my oxygen percentage is 19.64. Okay, could I take more data? Totally. Am I going to? Meh. I think I've got enough here. So once I've got my data, here's what I can do. I pre-populated, meaning I set up your graph for you to show time on the x-axis. Remember, we just talked about graphing, and my independent variable on the horizontal, across the bottom axis, is going to be time. Now, if I wanted to change that, I could just click on time, and I could select something else. But this is our independent variable. And now, I can choose, I'm doing this because I'm looking at it sideways, I can choose my dependent variable, or my y-axis. Now, I chose CO2 level, but you could also choose to look at the O2 level. Notice that it changes your scale for you to make it make some sense. But I'm going to look at CO2 level for now. So I've got this nice little zigzag, right? And my zigzag is corresponding to, at first, was it light or was it dark at the beginning when we started our experiment? Yeah, it was light. So as it's light, my CO2 looks like it's going down, and then it looks like it goes up during that dark period, and then it looks like it goes back down. My O2 level does the opposite. It goes up when it's light, down when it's dark, and back up when it's light again. All right, so I can only record for 15 minutes, so I'm going to pause here, and you're going to click on the next installment, part 2, to start talking about this and making some sense out of it.